Hello everyone, welcome back to the lecture 10 of NPTEL course Experimental Biochemistry, and today we will be talking about recombinant DNA technology. Okay, this course is coordinated by Professor Dasgupta and Professor De from IIT Kharagpur, and I am Beas Mukherjee, a research scholar from Dr. Shomode's lab, and today I will tell you about recombinant DNA technology. We will talk about the different processes, the different applications of RDT, and then we will look into the different steps, okay, of RDT. So what is basically RDT? It is also known as genetic engineering. So right now in today's world, this is a very relevant topic, and it finds a lot of different applications in different fields of science. So we will have a look at that, and finally we will be solving a lot of MCQ and MSQs, okay, based on whatever we discussed today. So let's start with the today's lecture. Right, so let us start talking about recombinant DNA technology. Recombinant DNA technology is also known as genetic engineering. So if you look at this figure over here, you will see that this is our DNA, of course you know, right? And here what they are doing is they are actually manipulating the DNA. Okay, so this is exactly what recombinant DNA technology is. It is manipulation of the genetic makeup of an organism. Okay, so there are different ways in which you can do that, and you can give desirable characteristics to that organism. You can maybe make that organism produce something, for example insulin, okay, which is a human growth hormone, but then it is produced inside other organisms, host organisms, right? So this is how we can actually engineer the DNA of that organism to produce this desired product in huge amount. Okay, so let's look into this. So recombinant DNA technology is also known as genetic engineering, and it is a set of molecular techniques used to manipulate and modify DNA sequences. So I'll share the screen and I'll go for slideshow. Okay, right. So this technology allows scientists to combine DNA molecules from different sources, creating a new genetic combination that do not occur naturally. Okay, so recombinant DNA technology has numerous applications in biotechnology, agriculture, medicine, and basic research. It is used to produce therapeutic proteins, develop genetically modified organisms, and with desired traits, create vaccines, study gene function, and more. So all of these different boxes that you see, they contain they are actually representing different field of science where these recombinant DNA technology methods are used to make all of these different things. So for example, better diagnostics. So this is in the clinical sector, okay. So therapeutics. See, there is better diagnostic tools, and then improved pharmaceuticals, personalized medicine. This is of very importance right now. Okay, this is a very high importance. So you can look up what is personalized medicine. This is basically exactly what kind of genes you have and what kind of diseases that can lead to. So you can alter your genetic makeup so that you do not get those diseases, okay, because certain mutations in our genes lead to different kinds of physiological complications, okay, that lead to the development of a particular disease. Then there is novel approaches to treat debilitating diseases and controlling pandemics. Then there is in the food technology sector, there is production of resistant crops, improving the yield, improving the shelf life of different food items that we eat, environment friendly fertilizers and biopesticides, and then there is the genetically modified plants, okay. So all of these are there. Then there is biofuel and biopolymer, food, recombinant proteins and vaccines, chemicals. Again, there is food, recombinant food is there, drug discovery, pharmaceuticals and resources for biomedical research, right? So all of these different things are different applications of recombinant DNA technology. You can have a look at all of these if you're interested. So this is a very interesting subject. All you need to do is just understand what it is about, and then it is going to be very clear to you. Okay, so let us try to understand what exactly happens in recombinant DNA technology. So if you look at this figure, this is actually a bacteria. Okay, so what does this bacteria do? This bacteria infects plants, and what they do is these bacteria leads to the formation of this swollen structures in the plants, and that is called tumor. Okay, and that is why the name of the bacteria is Agrobacterium tumefaciens. So it basically causes these tumors in plants. Okay, so now what is happening, and this tumor causing property is because of a particular plasmid that is present in the plant, that is called the tumor inducing plasmid or TI plasmid. So what humans have done is this plasmid, this gets transmitted very quickly. Okay, and because of that they have actually modified this. So they have used certain restriction site and they have cut this plasmid open. They have taken out all the genes responsible for the formation of all these different downstream processing, and all the genes that the plant maybe all the genes that the bacteria required. So all of those have been taken out, and the genes that we want the bacteria to have, so that it gives rise to plants with desired trait, that has been incorporated. This is called cloning. Okay, so see, this is the site where restriction enzymes have been used to cut open the DNA, double-stranded DNA, and then we are inserting the gene of interest that we want the particular plant to have. Maybe it is a gene for better resistant to um, other pathogens, or it is a gene for higher yield of crops. Okay, whatever the gene is. And then what happens, this plasmid is now called a recombinant plasmid. So what is recombinant plasmid? Recombinant means that its genetic material has been modified. That is the meaning of recombinant plasmid. Okay, now what happens, this is introduced into the, see, inserted tDNA carrying new gene. So that has been inserted, and then what happens? The plant is now getting a new trait. So this is used to treat the foreign DNA and plasmid with restriction enzyme and DNA ligase. DNA ligase is used for joining. Okay, restriction enzyme is used for cutting the DNA, and DNA ligase is joining with the newly introduced DNA of our interest. Then it will be introduced into the cultured plant cell and then regenerate new plant from the cultured cell. So this is what the entire process of RDT is. Now we will try to understand how this is done in greater details. Okay. So see, these are the different processes of recombinant DNA technology. So the first step is isolation. So DNA fragments containing the desired genes or genes are isolated from the source organism. Okay, so from the source organism you have to take out the specific gene that you want to amplify, and you want the target gene to be expressed in a different organism. So first that needs to be taken out. Then what will we do? We will cut that, right, using restriction endonucleases. So we will be learning about these restriction endonucleases soon. So restriction endonucleases, they are enzymes which recognize specific sequence of DNA, and they will go and cut those DNA sequences. Okay, so restriction endonucleases cut these DNA sequences and they produce fragments with sticky ends. Now what is sticky ends? If you look at this figure, you can see that this yellow fragment is exactly complementary to this region of the blue fragment. Again, this particular position also, it is the same case. So basically sticky ends are these single stranded extensions. Okay, here also if you see, these are the single stranded extension. So single stranded extensions are called sticky ends here, and blunt ends mean that both the strands are cut at the same place. Okay, this is a property of the different enzymes, restriction endonucleases, how they are exactly going to cut the DNA sequence. Fine. Then what will we do? So we will produce these sticky ends. For example, this is one strand and this is another strand. Here it ends. The next one is going to be like this. Okay, so for example, this is the strand of one DNA fragment. This is going to be the strand of your plasmid, into which you are going to put the DNA fragment. Fine, this is going to be exactly complementary to this one. Then we will insert this. So the DNA fragment containing the desired gene is inserted into a vector. This is done using DNA ligase, such as a plasmid or a viral genome. Okay. Then this vector is a molecule, it is a carrier molecule. Okay, this is going to carry the DNA of interest into the host organism. That is the vector. In this case, it is a plasmid. For example, this Ti plasmid, this is a vector in this case. Now, vectors can be different species. For example, they can be bacteriophage DNA, right? They can be your backmid, they can be your yak, and then they can be back. So they can be different, there can be different carrier molecules based on what is the size of your uh, desired DNA. Fine. Then what is done, the vector is often modified to introduce markers for selection and replication in the host cell. Now there is one important characteristic. How do we know that the vector got inside the host cell? So for that, the vector will be provided with certain selectable markers, okay, that are going to tell us that okay, this bacteria definitely contains the vector. Okay, besides that, the vector should also multiply, so that whenever this gene is expressed, it is produced in very high amount. The whatever gene the product is produced, that protein is produced in very high amount. So for that, it needs to have this ORI site so that it can multiply, which is the origin of replication. Okay, then there is transformation. So transformation, I'm sure you know, it is insertion of the plasmid DNA into the bacterial cell, right? If the same thing is done for your mammalian cell or any animal cell, then it is called transfection. Okay, so then see, bacteria, yeast and plant, it is called transformation, but for mammalian cells, for any animal cells, it is called transfection. Now selection. So host cells containing the desired recombinant DNA are identified and selected using markers included in the vector. Then there is expression. The recombinant gene is expressed in the host organism, allowing the production of proteins encoded by that gene. Fine. Now we will look at the different processes of RDT in greater details. You tell me if you are not understanding anything. Okay. I hope it is visible. Right. Okay. So let us start with this. So the first step of RDT is isolation. So isolation is, you have understood this, it is isolation of the desired DNA fragments, okay, from the source organism. So see, this is our desired DNA fragment which has been isolated from the source organism. Right, so this is the one, and this is the plasmid. Plasmid is containing your ORI site, it is containing multiple cloning site into which different restriction endonuclease cut sites are present, and inside that you are going to insert the gene of interest. Fine, and also there is different selectable marker which are going to tell us that yes, this recombinant plasmid is inside the bacteria. Fine. So this is how. So initially it was a plasmid, it was all natural, but then as soon as you modify the genetic makeup of the plasmid, it becomes a recombinant plasmid, right? It becomes recombinant DNA. Now what happens? What is DNA cloning? So DNA cloning is the process of making multiple identical copies of a particular piece of DNA. Okay, in a typical DNA cloning procedure, the gene or the other DNA fragment of interest, perhaps a gene for a medically important human protein, is first inserted into the circular piece of DNA called the plasmid. This insertion is done using the cut and paste DNA. It is done using basically your restriction endonucleases and DNA ligases, and then a molecule of recombinant DNA is produced and DNA is assembled out of the fragments from multiple sources. Fine. Now we will see what happens next. So this figure tells you how exactly the restriction enzymes work. Okay, so right now what you have is you have this gene of interest. In the last class we studied exactly how DNA is extracted from different organisms that we have studied. So exactly following that way you are going to extract the DNA of interest from that organism. These days it can even be synthesized. Okay, gene synthesis is possible, so it can be synthesized. And then what happens, this is a restriction enzyme. So restriction enzyme, this is GAATTC, so this is the cut site for EcoR1. That particular restriction enzyme is going to identify this sequence, and if you read the sequence from 5 prime to 3 prime and 3 prime to 5 prime, it is reading the same, right? And therefore it is called a palindromic sequence. So this restriction enzyme is going to identify this palindromic sequence. It is going to cut here and it is going to cut here. So what happens, you get this single stranded overhang here and another single stranded overhang here. So this tells you that these are basically sticky ends, right? And then we take the DNA of interest. Inside the DNA of interest we introduce this restriction enzyme cut site, and it is cut with the same restriction enzyme. After that what happens, these two are ligated. So this initially the molecule was blue, and this is your DNA of interest, which is pink, and then you see DNA ligase joins them and it becomes a continuous strand of DNA. Fine. But right now what is done is we do not use the same restriction enzyme for both the ends. Okay, we use two different restriction enzymes, and so that means for one full vector, if you look at this, okay, someone is trying to join. Slide number 6 is not visible. Okay, 1 second. It is now visible. All right. Okay, you tell me if it is not visible. I will slow down. Okay. So Srishti is finally speaking. Okay, very good. You tell me, okay, communicate. Okay, then that will help you a lot. Okay, so we'll continue. Okay, so if you look here, in that case I will not be going for slideshow. Okay, because in slideshow a lot of times there is a delay. So in that case we will just go like this, if that is okay. Okay, so let's carry on. Fine. So So now there is a very interesting concept that is called the restriction modification system in bacteria. So what happens here is, see, bacteria is having its own defense mechanism. Bacteria is attacked by viruses. Now these viruses are called bacteriophages. Okay, so bacteria is having their own defense mechanism. So what happens there? Bacteria, what it does is it methylates its own DNA. So it adds methyl group to the bases. For example, there is A gets methylated, C gets methylated, right? So that is how bacteria methylates its own DNA using the methylase enzymes. And the DNA that is coming from any foreign source, for example, any bacteriophage, okay, that is not methylated because bacteriophage is not having any enzyme of its own to methylate the DNA. And therefore what happens, that DNA is recognized by the bacteria as a foreign DNA. Then what happens, bacterial restriction endonucleases, they go and cleave that foreign DNA. This is how the restriction modification system in bacteria works. Now this is for the defense of the bacteria. Fine. So let's see what happens here. The restriction modification or RM system is a defense mechanism found in bacteria and archaea that helps protect the host organism from foreign DNA, such as viral DNA or bacteriophages or DNA from other sources. It consists of two major components. One is your restriction enzyme. These enzymes recognize, as I told you, specific DNA sequences and typically short palindromic sequences. So palindromic sequences are the ones that read exactly the same whether you read it from 5 prime to 3 prime or the 3 prime to 5 prime of the other strand. So these are palindromic sequences and they cleave the DNA near these sequences. Fine. Then comes the modification enzyme. So these enzymes methylate the host DNA. Host means it is bacteria over here. So these are methylating the bacterial DNA, and what they are doing is they are at the specific recognition site. So even for methylation there is specific recognition site and it is protecting from cleavage by corresponding restriction enzymes. So the modification enzymes recognize the same sequence as the restriction enzymes but methylate it, preventing the uh, host's own DNA from being cut. So this property is basically of the type 2 restriction enzymes. Okay, now restriction modification system there are different types of enzymes in the restriction modification system. There are 4 different types of enzymes. So the ones that we use in the laboratory most, they are the type 2 restriction enzymes. Now very soon we will find out why that is the case. Now if you look at this slide, you will know that the restriction modification system works as follows. The modification enzyme methylates the host DNA at specific recognition site, protecting it from cleavage. When the foreign DNA such as viral DNA enters the host cell, it is not methylated and lacks protection. The restriction enzymes recognize the unmethylated foreign DNA and cleave it at specific sites, rendering it non-functional. Right, so this is once again a defense mechanism against invading foreign DNA such as viruses. Fine. Now if you look at this chart, you will see that these are the different features and these are the different characteristics of the 4 different types of restriction enzymes. So first is subunit. So type 1 is very big. It is having 3 different subunits. Type 2, 3 and 4, they are having only 2 subunits. One is endonuclease, the other one is methylase. Fine. Then that is the case for type 2. And for type 3, there is endonuclease, methyltransferase and ATPase. And here also it is endonuclease and GTPase. Right. Then there is different cofactors for DNA. So it requires, you know that enzymes require certain kind of cofactors for their activity. So for type 1, it is ATP, it is Mg2 plus. For type 2, it is only Mg2 plus. Right, and that is why whenever you set these restriction digestion, these reactions even in the laboratory, you have to add buffer containing Mg2 plus, otherwise it will not work. Right. Then there is ATP Mg2 plus given in type 3 enzymes, and for type 4 enzyme Mg2 plus and GTP is required. Right. Then there is methylation. So methylation happens, and in case of type 1, the recognition site is asymmetric. Okay, so what happens? The recognition site and the cleavage site are very separated around 1 kb. Okay, and then for type 2, the recognition site is the cleavage site. For the others also, recognition site and cleavage site are very far away. And that is why it is very difficult to know exactly where the restriction endonuclease is going to cut. And therefore, if you have your gene of interest, maybe it is going and cutting at the gene of interest. You never know, right? Even though the recognition site is very far away. So fine. Then there is DNA translocation, and for all of these, DNA translocation is required, but for type 2, DNA translocation is not required. Now again, we are doing the reactions in vitro, so this is not going to matter to us. Fine. Then finally, we will look at the different enzymes used in recombinant DNA technology. So the first enzyme, I am sure you have understood, that is restriction endonuclease. That is one very important enzyme which is used to cut the DNA of interest and cut the plasmid into which it is going to get incorporated. Now who is going to join them? So for joining, DNA ligase is the enzyme, okay, which joins the 2 different DNA fragments. So DNA ligase is an enzyme that plays a crucial role in DNA replication, repair and recombination process. Its main function is to catalyze the formation of phosphodiester bonds between adjacent nucleotides in the DNA strand. So, sealing nicks. Nicks are single-stranded breaks in the DNA in the sugar phosphate backbone, and this process is essential for joining DNA fragments together during DNA replication. The repair of DNA damage and creation of recombinant DNA molecules is performed by DNA ligase. Fine. Now let us see how DNA ligase is going to do all of this work. So if you look at those four steps, okay, those four are the steps involved in its mode of action. Okay, so the first one is adenylation. So what happens in adenylation? There is ATP. ATP is required as a cofactor. From that ATP, AMP group is actually attached to the, see here, to the substrate. So basically DNA ligase first catalyzes the adenylation of itself using ATP. So AMP group is attached to a lysine residue within the ligase enzyme. So inside the ligase enzyme at the active site there is a lysine residue. To that lysine residue AMP is adenosine monophosphate, right? That is going to get attached. Then what happens? Then there is binding. So now what happens? The ligase enzyme then binds to the nick. Nick is a single-stranded break in the DNA or gap in the DNA double helix where ligation needs to happen. Okay, so transfer of AMP, the enzyme goes and binds, and then there is a transfer of AMP from the ligase enzyme to the 5' phosphate group of DNA that contains the break. Fine. Then there is a ligation reaction. What happens in the ligation reaction? There is a nucleophilic attack of the 3' OH. So ligase catalyzes the formation of a phosphodiester bond between the 5' phosphate and the 3' OH groups of adjacent nucleotides that are sitting side by side, sealing the nick in the DNA backbone. This is how it joins. Okay. We will look at the figure to understand this better. So if you see here, the entire reaction has been given. Now questions are asked from DNA ligase. Okay, and then we will look into the different types of DNA ligases. So here this is very important. They might ask you exactly which residue in DNA ligase is involved in this entire ligation process. So for that you have to say that it is a lysine residue. So this is a very important concept. Okay, so if you look at the figure there is DNA ligase, and in the DNA ligase there is this lysine residue, and to the lysine residue, to the epsilon amino group of the lysine residue, AMP is going to go and bind. Okay, AMP from where is it coming? It is coming from ATP, because ATP is used in this reaction. Okay, so ATP gives AMP that goes and binds. Then what happens? The enzyme goes and binds to the 5' end. Excuse me. So the entire enzyme goes and binds to the 5' end, and then to the 5' phosphate this AMP group is transferred. Then what happens? There is a nucleophilic attack from the 3' OH, and if you see the reaction here, the 5' phosphate plus AMP ligase complex forms DNA 5' adenylated plus ligase with AMP. Then what happens? Okay, so we will carry on. Then what happens is, this DNA, which is 5' adenylated plus the 3' hydroxyl group of DNA, both of them join using this phosphodiester linkage, and AMP is released in the process. So this tells you that this entire process requires ATP, right? Okay. So we will go on to the next slide. Now this slide tells you the different kinds of ligases that are mostly used. Okay, so usually two different forms of ligases are used. One is T4 DNA ligase and another one is E. coli DNA ligase. So T4 DNA ligase is derived from this T4 bacteriophage. This is more commonly used in molecular biology laboratories. Why is that? because this requires ATP as its cofactor, but E. coli DNA ligase requires NAD as the cofactor. Okay, and then you can see that substrate specificity. So basically this E. coli DNA ligase usually seals blunt-ended DNA molecules. So blunt end means that there is no single-stranded overhangs. But then for T4 DNA ligase, this prefers your single-stranded overhang. So then if a DNA sequence has been cut with EcoR1, you will have to go for T4 DNA ligase. Okay, you cannot go for E. coli DNA ligase in that case. Then again, E. coli DNA ligase requires pre-existing 5' phosphate and 3' hydroxyl on both the strands, and T4 DNA ligase can join mix in DNA without pre-existing 5' phosphate. Fine. And then the reaction conditions, you can see that T4 DNA ligase requires sticky end ligation. Fine. There is one very interesting thing here, the temperature, optimum temperature. So optimal temperature for E. coli DNA ligase is 37 degrees centigrade. For T4 DNA ligase, it happens at 37 degrees centigrade as well, but then it actually opens. Okay, so for it to happen properly, you need to go for a lower temperature. So it is a compromise between the good ligation reaction and the optimal activity of the enzyme. Now we will move on to the next slide. So the next thing is, so you have taken this DNA molecule, DNA of interest, you have put that into the vector, you have ligated it with the vector. So this is one whole single molecule now. Now you want this entire plasmid to get into the bacteria where it is going to express, right? So how do you put the plasmid into bacteria? So first thing is, you have to choose a bacterial strain which is compatible and it is going to uptake this plasmid. The second thing is, you need to have a proper method using which you are going to introduce this plasmid into the bacteria. So there are different methods for that. The first thing is choosing a competent cell. So what is a competent cell? Competent cell is a cell which is capable of taking up DNA, foreign DNA from outside. Okay, and that is called a competent cell. So how can you make a cell competent? You can go for different chemical and physical methods. So competent cell are having certain membrane characteristics. You have to look into that in greater details. Since we do not have questions on this, I have not covered that portion. So you need to look into that in greater details, exactly what proteins, what membrane properties make a cell a competent cell. Okay. So this is the chemical method that they are talking about here. This is the chemical method of transformation, and then there are also different physical methods such as electroporation. So in electroporation, what happens? There is an electric shock is given at a specific voltage for a specific amount of time, okay, some specific milliseconds, and then what happens? The membrane, it becomes very porous. Okay, because of this electric shock, the membrane becomes porous, and through that pore, the vector actually goes in, the plasmid goes in. That is called electroporation. There is micro injection, there is gene gun. Okay, all of these are used for your mammalian cells. Gene gun is used for plant cells also. So all of these different kind of methods are there. You can have a look at those methods as well. Okay, and then the most commonly used method is chemical transformation. In this, what happens? Heat shock is given. So the bacterial cell, it is usually kept in ice. Okay, it is taken out from -80 degrees centigrade refrigerator where it is stored, and then it is taken out, it is placed in a ice, and then what happens? From ice, that is temperature is nearly 0 degree. So from ice, you place it into a water bath with temperature around 42 degrees centigrade. So in 42 degrees centigrade, you keep it for 30 to 40 seconds. Then what happens? You take it out and you put it into ice again. This gives a shock to the bacteria. And what happens? The membranes in the bacteria, it becomes porous. Once again, the plasmid can go in. If you look at the figure, you can see that initially the membrane was intact, and then it has become porous after the entire heat shock procedure. And so now the plasmid Here the plasmid is shown as round, but originally plasmid is a supercoiled molecule. Okay, and so it is even easier for the plasmid to get in. Fine, and then finally what happens? There is recovery. The cell membrane becomes intact again. So let us see what is given in the slide. So transformation and competence. Transformation is a process in molecular biology where foreign DNA is introduced into a bacterial cell or other cells. A competent cell refers to a bacterial cell that has been treated to increase its ability to take up foreign DNA during transformation. So competent cells are typically prepared by subjecting bacterial cell to certain environmental conditions or chemical treatments that disrupt the cell membrane and allow the foreign DNA to enter into the cell more easily. Right? So we have even introduced the DNA of interest into our bacteria. Now what happens? Now all the bacteria, they are going to grow. You have transformed the bacteria, you have plated the bacteria on a agar plate, and you see that there are tiny colonies. You come the next day, you see tiny colonies. Now how are you going to know which bacterial colony is containing your recombinant plasmid? So for that, when we are making these agar plates, agar is basically, it is making the media semi-solid, right? It is making it solid. And LB or Luria Bertini media, that is actually the food for bacteria. So along with the food, you are also adding some antibiotic resistance molecule into it. Sorry, you are adding an antibiotic into it, right? So it can be ampicillin, it can be kanamycin, it can be chloramphenicol, any antibiotic you add. Then what happens? Antibiotic is going to kill the bacteria. Now inside the plasmid DNA, there is going to be this antibiotic resistance gene. So you can see this pink part of the plasmid, that is the antibiotic resistance gene. That gene is also going to get expressed, okay, and it is going to ensure that whatever antibiotic is there in the media, the bacteria is equipped to fight that and it survives. So only the colonies that grow on the plate, all of them are having this antibiotic resistance gene. Now here comes two different terms. One is a transformant, another one is a recombinant. So transformant is a bacterial cell which has taken up the plasmid. Okay, what is a recombinant? Recombinant is a bacterial cell which has taken up the plasmid containing your desired DNA. Okay, so all the cells that are growing, they are all transformants. But then are they recombinant? Has the genetic material of that particular bacteria been altered in any way? So that you need to find out. Okay, so see, look here. The plasmid typically contains an antibiotic resistance gene which allows the bacteria to survive in the presence of a specific antibiotic. Thus, bacteria that took up the plasmid can be selected or nutrient plates containing the antibiotic. So bacteria without a plasmid will die and the bacteria carrying a plasmid can live and reproduce. Each surviving bacteria will give rise to a small dot-like group or a colony of identical bacteria that carry the same plasmid. So bacteria without a plasmid are going to die and the bacteria with the plasmid makes a colony. We'll go to the next slide and we will try to understand what exactly is happening here. Okay. So see, what is the role of ampicillin resistance and LAC-Z gene? So if you remember LAC-Z gene, LAC-Z was there in the lac operon, right? So LAC-Z. Okay, so let us have a look. See, there is the bacterial plasmid. Bacterial plasmid is having your ampicillin resistance gene and it is having this LAC-Z gene. LAC-Z gene gives rise to lactose. Okay, sorry, it gives rise to beta lactamase. So, beta galactosidase. So, beta gal, right? So, LAC-Z gene is giving rise to your beta galactosidase. So, beta galactosidase is going to act on galactoside it is going to act on lactose and it is going to break down into your glucose and galactose, right? So beta gal is going to act on lactose, break it down into glucose and galactose. Now this entire plasmid has been modified in such a way that uh, if you see here, yes, so initially what is done, the plasmid or the vector plasmid is isolated and the DNA, that is the human DNA of interest, that is taken. So see, human DNA of interest has been taken and this plasmid has been taken, and then what happens, the sticky ends are produced using the same restriction enzyme for the end of the, for one end of the DNA of interest and one end of the plasmid DNA. Both of them are ligated using DNA ligase and they are joined by base pairing. Some of the plasmids like this one join with the gene of interest. So they are joined. Now this is a recombinant plasmid. Then what happens, at DNA ligase to bond covalently and human DNA containing the gene of interest plus the non-functional LAGZ gene. So LAGZ gene, it has been a certain portion of it has been deleted. Okay, so now LAGZ gene is producing only one particular subunit of this beta galactosidase, and the rest of the portion of the LAGZ gene, that has been introduced into the genome of the bacteria, and so the rest of it is coming from the genome of the bacteria. So there is an alpha and there is an omega portion. So both of them join together to give you the functional beta lactamase, and only when the beta lactamase is functional, the bacteria is going to survive. So put the plasmid into LAGZ minus bacteria by transformation. So this bacteria is not having any gene for beta lactamase, and then what happens, this is becoming functional. So clone the bacterial colony, clone cells, plate cells onto media with ampicillin and X-gal. So X-gal is the substrate for beta lactamase. This is a chemical substrate, and identify clones of the cells containing recombinant plasmid by their ability to grow in the presence of ampicillin and their white color. So this concept of blue-white screening. So blue-white screening is a very important concept. So what happens is, when the bacteria is growing and it is converting this beta gal into a blue color compound, okay, then what is happening, it is having functional LAGZ gene. So that is giving blue colony. But when the LAGZ gene is non-functional, because inside the LAGZ gene the multiple cloning site has been introduced. Multiple cloning site has different sequence of different restriction enzymes. Then what happens, we are introducing the DNA of the DNA insert inside that LAGZ gene. So what is happening, a very important phenomena, this is called insertional inactivation of the LAGZ gene. So because of insertion of the DNA of interest, LAGZ gene is getting non-functional. So that is insertional inactivation of LAGZ gene. Now what happens, even if you add X-gal, no blue product is formed because beta galactoside is not getting produced. So that is giving you white colonies, that is talked about here, right? So this is called blue-white screening. If it is a recombinant plasmid, it is a recombinant cell population, it is going to give you white colored colonies. If it is non-recombinant, it is going to give you blue color colony. Okay, I'm sure you've understood that. So cloning DNA into bacterial plasmid allows the bacteria to serve as a factory for making large quantities of plasmid of interest. Now you can ask me questions by typing in the chat box. Okay, I'll answer, or you can even switch on your mic and you can ask me questions if you want. Now, so we have inserted this DNA of interest inside the bacteria. Now what is going to happen? So if you look at this, now here actually we are going to talk about recombinant protein. Okay, let's say that this particular DNA of interest codes for a particular protein. Now see, let's see what happens. So this is this DE3. Now we had talked about this BL21 cell in our previous lectures, where we had talked that this particular DE3 segment of the DNA, this is an engineered portion taken from lambda DE3 plasmid. Lambda DE3 bacteriophage, right? So this contains T7 RNA polymerase promoter, which is going to T7 RNA polymerase gene, which is going to code for your T7 RNA polymerase enzyme. Now what happens, and for just before this particular gene there is a lac promoter. Okay, so this lac promoter, you can see this P here, lac promoter is very modified. So this is lac UV5, in which what happens, there are certain modifications, and this promoter is, now this modification is done because lac promoter is a leaky promoter, and we want to make sure that even not basal level of this T7 RNA polymerase is produced. So we need to ensure that, and for that those mutations have been done. Then there is lac operator. Okay, so I'm sure you remembered this entire concept of lac operon. What happens is, RNA polymerase comes and sits on the promoter, it transcribes this T7 RNA polymerase when you are ensuring that lac operator is not being guarded by the lac repressor. Okay, and that is why IPTG is added. Then what happens, this RNA polymerase, T7 RNA polymerase gets produced, and the vector that we are providing, vector for protein expression, let's say it is PET vector, so it is having this promoter for T7 RNA polymerase. T7 RNA polymerase goes and sits on the promoter, and next to the promoter your gene of interest is present. So this is a viral DNA promoter, and what happens, a lot of uh, translation happens, and a lot of protein gets produced. You can see this is the protein, this is called overexpression, okay, of the protein, and that is exactly what we are seeing here. So this is how recombinant protein is produced in the bacteria. Recombinant protein is overexpressed. Fine. Now we will look at gene expression and gene expression and recombinant protein. Okay, we will just look at the theory portion. Now I am sure you've understood exactly what happens. We look at the theory part. So once we have found a bacterial colony with the right plasmid, so how do we come to know it is having the right plasmid? We need to extract the plasmid, and we need to sequence the plasmid. So DNA sequencing, Sanger sequencing, that needs to be done. Okay, so DNA sequencing has to be done, and we can grow a large culture of plasmid bearing bacteria, and then we give the bacteria a chemical signal. So this chemical signal is your IPTG, right? So IPTG, we had learnt about this in the previous classes when we were talking about protein expression and characterization. So see, a colony is taken and that is grown into a large culture. Bacteria are induced to express the gene of interest, and then what happens, it is produced and mRNA is translated into protein, and that protein is a recombinant protein, which can be insulin in this case, right? So we get large quantities of protein from bacteria. When the protein is not even bacterial protein, it is human protein or any other source, right? So let us start with the questions now. Okay, so this is week 10 assignment. All questions are there. You can answer in the chat box, and you can switch on your mic and you can tell me the answers. So these are the first two questions. Let's see who can answer them. So in the first question, they are asking you what ion is used to make sure that the recombinant plasmid gets into bacterial cells. Okay. So let's see who can answer. Okay. So you're saying it's 1C. Yes. Very good. Okay. Very good. So Srishti is also answering. Very good. And let's see. What about Naveen? You can answer too. Okay, so what about the second question? The second question, to join two different DNA fragments. Okay, so that is A. So to join two different DNA fragments, which of the following enzymes is used? DNA ligase. Very good. Right, that is the right answer. So you can look here at the answer. So basically competent cells are treated with divalent cation, specifically calcium. Okay, and what happens is the pore size increases. Now what happens here is that these divalent cations, calcium ions are very big, right? And the DNA that you are adding, DNA is basically negatively charged, right? So it is whatever plasmid DNA or any DNA that you are adding, it is basically negatively charged. The phospholipid bilayer, or let's say the LPS, right, of the bacteria, whatever cell membrane is there, whatever cell wall is there in the bacteria, lipopolysaccharide, it is also negatively charged. Then what happens? It is going to repel. But this divalent cations that you are adding, these divalent cations, they are forming a bridge, and it is neutralizing the uh, plasmid DNA's negative charge. That is why it is facilitating it to take it out, right? But why exactly calcium and why not magnesium, that you need to find out. You need to look into the entire cell morphology, the characteristics of the membrane proteins of competent cell to learn exactly why calcium is used and why not magnesium or manganese is used, because these are also divalent, right? So for the next question it is, right, so it is definitely ligase molecule. So let us look at the next one. So the process by which a plasmid or vector is inserted into the host cell, what is it? Yes, very good. It is transformation. Then there is a match the following. So see, gene of interest. Okay, I can already find the right answer. What about a plasmid? Plasmid. Okay, it is what about a clone? Okay. So what is the answer for the fourth question? You're saying it's 4D. Okay, let us see. 4 is A. Why is it A? Let us see. See, for gene of interest, it is a DNA segment that is to be inserted or deleted. Right. Plasmid. Plasmid is a small circular double-stranded DNA molecule that is distinct from the cell's chromosomal DNA. Plasmid is not genetically modified. Okay, plasmid has not been touched by humans. Remember that plasmid can be made vector when it has been artificially modified. So see, DNA molecule used as a vehicle to artificially carry foreign genetic material into another cell where it can be well expressed. That is your vector. That is the only difference between a plasmid and vector. Plasmid has not been touched by humans. Vector has been genetically modified by humans. Okay. Then there is clone. What is a clone? Clone is a organism that carries identical genome. They are called clones. So that gives you this entire option A as correct. Now question number 5 and 6. Try to solve them. In PCR, magnesium cations are used. Why is that so? PCR is your polymerase chain reaction in which DNA is amplified. Okay. So why is magnesium used? So you are saying it's B. It is a cofactor of the enzymes used in PCR. Yes, that is true. So what are the enzymes used in PCR? Excuse me. For the next question, it is what is the role of selectable marker in a cloning vector? Okay, so maintaining the vector in the cells, selective survival of the cells that have the vector, increasing the cell growth, and okay, yes, that is correct. B is the right answer, selective survival in the cells. Now what enzyme is used in PCR? Polymerase chain reaction. So DNA polymerase is used. Now polymerase from a very specific bacteria is taken which is a very thermostable polymerase, right? So Taq polymerase is used, modified versions of Taq are used. Basically DNA polymerase is used in polymerase chain reaction. Fine, now we will go to the next question. Okay, so here are three different questions. So see, what does the first question ask? Multiple cloning site in a vector has what? Correct, unique restriction sites. Very good. Which of the following should be present in a vector? Cloning site, antibiotic resistant gene, and origin of replication. So the answer should be D. Correct. So all of these should be present. Right? It needs to have a selectable marker, which is your antibiotic resistance gene. It should have an origin of replication so that it can multiply. Otherwise, there will be no protein overexpression, right? And it should have a cloning site into which we can insert our DNA of interest. Then what will happen if the origin of replication, I told you the answer here. So what will happen if the origin of replication is missing in a vector? So vector will not be taken up by the cell. Vector will not contain the gene of interest. The vector will not survive in the cell. Vector will not replicate. What is going to happen if origin of replication is not there? That is right. Vector is not going to replicate, right? Okay. So those are the answers. You are very correct. All of you are correct. Right, so now we will look at a DNA sequence. Now these kind of questions get asked in your exams, but a more difficult version of it gets asked. Okay, here it is a very simple question. So they have given you a DNA sequence and they are asking you exactly which one of these enzymes can be used for cutting this DNA sequence. Okay, so for that you need to find the exact those 6 base sequence over here. Okay, let's see. Yes, very good. It is having this particular sequence here, and so NDE1 can be used. So a restriction enzyme is an enzyme that cleaves DNA into fragments at or near the specific recognition site within the molecule. Okay, so it should be the second option. Now we will go to the next slide. So which one of this is true? So the first option is DNTP mix consists of a mixture of restriction enzymes and primers. Exponential amplification of a gene can be done for a very small amount of DNA. Thermostable DNA polymerase is absolutely necessary for PCR. Antibiotic resistance is achieved through the use of selectable markers. So which one do you think would be the right answer? So you are saying it's D. DNTP mix, yes, very true. DNTP mix does not contain restriction enzymes and primers. This is deoxyribonucleotide, right? So deoxynucleotide triphosphate. So it is only going to have ATP, GTP, CTP and TTP, right? Amplification, exponential amplification, yes. Your PCR reaction is there. So 2 to the power n. After every cycle, if there are 40 cycles, you get 2 to the power 40 different molecules, right? So that is exponential amplification. Thermostable DNA polymerase is absolutely necessary for PCR. That is true because in PCR we repeatedly heat the DNA to denaturable temperature. So that is 98 degrees centigrade. We heat the DNA to that high temperature. If the polymerase is not thermostable, what is going to happen? It is going to get degraded. So then after every step you need to add the polymerase, and that would be very costly, because enzymes are very costly, right? So after every step, if you are adding fresh enzyme, that is going to cost you a lot. Fine. So what is the last one? Last one, I'm sure you've understood that yes, it is a antibiotic resistance is a very well accepted selectable marker. So if you look at this figure, you can see that the buffer is there, and tag DNA polymerase, this is added, DNA template is added, and nucleotides and primers, all of these different components are added, and only then the PCR reaction happens. So DNTP is a mixture of ATP, GTP, CTP and TTP. This should be TTP, shouldn't be DTP, right? Okay, so we will go on. Right, so this slide tells you more about PCR. You can look here. So what happens, what are the different steps in PCR? So PCR is polymerase chain reaction. This is a molecular biology technique used to amplify a specific segment of DNA. It involves a series of temperature cycles that enable the selective amplification of the target DNA sequence. What happens in PCR? The first step is denaturation. So both the strands of DNA are here and both the strands get separated. And to each strand you get the primers which come and bind, and then the primer gets extended. So primer binding is the annealing step. Extension of the primer, complementary bases are added to the DNA based on the sequence of the DNA. That is your extension steps. So for the enzyme used, the extension step is 72. So that means the optimal activity for Taq polymerase is at 72 degrees centigrade. So you can imagine, right? So this particular enzyme has been taken from a bacteria that stayed in hot springs, right? So in hot spring the temperature is very high, and therefore its optimal activity is at such a high temperature. Okay, now finally what happens is, right, so there are more questions on PCR. Try to solve them. What is the correct sequence for the steps of PCR? Denaturation, annealing, extension and hold. Okay, very good. And then which of the following is not a stop codon? D. Correct. Why is that so? Because ATG is the start codon, right? Others are stop codons. Correct. So yes, denaturation, annealing, extension and hold. So at what temperature is hold done? Do you know? Temperature at which hold is carried out. No. Hold is done at 4 degrees centigrade or 0 degrees centigrade. So it is equivalent to keeping in ice. We want the reaction to stop, right? So that is why we want to make the enzyme and we want to make the DNA and everybody non-functional, relaxed. So we are keeping them in ice. So hold is basically 4 degree or 0 degree centigrade. Okay, now we'll go to the next one. So see, try here. Correct. Now how do you decide this annealing temperature? You can type in the chat box, you can tell me whatever is your choice. How do you decide the annealing temperature? So see the first one, that is 95 degrees centigrade, that is the temperature where denaturation is performed, right? Both the strands are separated. Then what happens, the primer comes and binds, right? So at what temperature is the primer going to bind? How do you decide that? 60, yes. But then I'll tell you how exactly that is decided. So basically what happens is, when you design a primer sequence, now primer sequences you design, okay, using website, you manually also you can do. When you are taking that and putting it on the website, then what happens, they tell you that this is the GC content. So what is GC content? It is the amount of G or C present. So you know that if G or C is present in the primer, it is going to bind to the complementary DNA sequence very tightly, because there are um, three your hydrogen bonds, right? So if the GC content is very high, the primer is going to have a very high melting temperature. So what is melting temperature? Melting temperature is the temperature at which half of the entire primer molecules get dissociated from the DNA, complementary DNA, right? So based on the melting temperature of the primer, you decide this annealing temperature. So annealing temperature is usually kept 5 degree less than the melting temperature for the two primers. So let's say your one primer is having a melting temperature of, let's say, 72 degrees centigrade. The other is having a melting temperature of 70 degrees centigrade. Okay, then in this case your annealing temperature is going to be 5 degrees less than the one with the lower melting temperature. So that is 65 degrees centigrade. So at 65 degrees centigrade, you are going to see both of them bind to DNA and remain there. So that is how you decide this annealing temperature. So higher the difference between the Tm of the primer and the annealing temperature, more specific is the reaction. But if you don't want the reaction to be very specific, let's say there is some kind of mutation that is to be performed, okay, mutagenesis is to be done, then what do? You design mutations in the primer, but then the DNA sequence is not having any mutation, right? Then what you do, you make sure that the primer comes and binds even though there are certain mutations. Then what do? Is you increase this gap between the annealing temperature and the uh, melting temperature. Then what happens? More non-specific binding happens. Amongst those non-specific binding, maybe one of those is your desired product. So that is how we play around with specificity and temperature. Okay. So what about the next question? See, which of the following questions is true for heat shock method? This method is used to kill bacterial growth. Okay. Heat shock, the name definitely suggests something like that. So it increases the cell permeability of bacterial cell. In this process, competent cells were incubated at 42 degrees centigrade for 45 minutes. Okay, be very careful. C is not correct because it is 45 seconds, not 45 minutes. Okay, and so yes, only B is the right option, right answer. So yes, it increases the cell permeability for bacterial cell and it is incubated at 42 degrees centigrade but for 45 seconds. Okay, and that is the end of the chapter. So you can refer to these two books. There is Primrose and there is also T.A. Brown. Those two books you can refer to. Those are good for for molecular biology. Okay, and there are many other books on RDT. There are so many different books on RDT. So RDT is a very interesting subject if you understand it. And you can reach out and you can also go through this different questions. There are different websites provided. So thank you so much for attending the class. If you have any questions, you can ask. Double digestion step. double digestion. So are you talking about restriction digestion? Okay, sure. So, right. So what happens in double digestion? So let's say if I explain to you here. So let's say that initially this is your, okay, this is not working correctly, but I hope you can understand. So let's say that this is your plasmid DNA, okay, and you want to integrate another DNA of interest. Let's say this is the DNA of interest that you want to insert into this plasmid DNA. Now what we do is, we want to now, one second. So let's say this is the plasmid DNA. It is having two different strands, okay, and with the plasmid DNA you want to join, let's say, this particular small DNA insert, okay. Now what do you do? Now this plasmid DNA is containing this multiple cloning site, which is your MCS. Okay, so it is having this MCS. Now what you do is, for this one side of the MCS, you have one particular restriction enzyme. Let's say it is R1. Okay, let's say one end of the MCS you cleave with eco R1. Let's say another end of the MCS you cleave with BAM H1. Similarly, for this insert, this side you go for your restriction enzyme 1, which is eco R1, and for this side you go for BAM H1. Then what happens? Two distinct type of so two distinct type of sticky ends will be produced. For this case, let's say that this is producing this kind of a sticky end, and for this one the sticky end is normal, okay, it's like this, sticky end is normal. Similarly, for this one also you will get two different kinds of sticky ends. For this part you will have a sticky end like this, and for this part you will have a sticky end like this. And that is why what happens when you join this portion and this portion is complementary, and this portion and this portion is complementary. This ensures that this entire gene is not inserted in an inverse manner, okay, because when the protein will be formed, if it is inserted in an inverse manner, then you will not get the desired product. So exactly, that is why two distinct restriction enzymes are used, and the entire process is your double digestion. So two different restriction enzymes. Oh, it is not showing. Did you not see any of the visuals? Okay. Okay. I will try to explain without the visual. Okay, 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 fine. So try to understand. Let's say this is the plasmid. Okay, let's say this is the entire plasmid, and here is your multiple cloning site, that is the MCS. Now you want to cut open this MCS, and here you want to insert the DNA of interest. Let's say this big thing is your DNA of interest. Now what you do, this portion of the MCS, this end of the MCS, you cleave with one particular restriction enzyme. Let's say it is EcoR1. And here, this also you cleave with one particular restriction enzyme, which is EcoR1. So what happens? This produces a particular type of overhang. Sticky ends are going to get formed. This is the sticky end, and here this becomes a sticky end. So this is how it is exactly going to fit in. What happens to the other side? This portion of the vector you cleave with another enzyme. Let's say it is your let's say it is your BAMH1. So it is going to have this kind of a different kind of a sticky end. Again, your DNA of interest, you are going to cleave with a different kind of restriction enzyme, which is BAMH1, and so it is going to have a complementary sticky end, and it is going to basically bind to this. So that ensures that if this is your DNA of interest, let's say this is your DNA of interest, it is cleaved with two different restriction enzymes. Like this pen, it is having nib in one side, and like the different side is different, right? So it will ensure that it is not inserted in this orientation, because if it gets inserted in the opposite orientation, you are not going to get the specific protein product that you want, because your start codon is going to be here, promoter is going to be here, there is going to be the stop codon, so no protein comes, correct? So it has to be inserted in the right orientation. That is why two different restriction enzymes are used. So did you understand? Okay, great. Do you have any more questions? You can ask me if you want. Thank you so much. Thank you for attending the class. Thank you so much.